Good evening, everyone. Hope I'm audible. Hello, dear students. Just let me open this thing, your YouTube chat, so that I can respond to your comments. Good evening, everyone. Hope I'm audible. So, just let me confirm once if I'm audible or not, so that we can continue. So, in this today's session, we are going to do about biochemistry. So, just let me confirm once if I'm audible. Okay, perfect. So, good evening to your students. Welcome to today's session. Actually, the session had to be postponed due to some personal issues. I could not manage the time. So that's why it has got a little bit changes in the schedule, but it doesn't mean that I will not do the session. So anyways, we are here. So let's start off with our discussion. So in biochemistry, as I have told you people, there are a lot of important topics. In that very most important topic from the biochemistry is going to be our carbohydrate chapter, because carbohydrate has a lot of MCQs. Okay. All right. So when we are speaking of carbohydrate, let's go to the first and most important concept, that is classification of carbohydrate. So carbohydrates are classified into multiple types. They're called as monosaccharide. Monosaccharides are a type of sugars which are a type of called as monomers of the carbohydrate. What do I mean by that? Monomer. So what is a monomer of a carbohydrate? For example, I have a monomer that is named as a glucose. Named as a glucose. So if I break this glucose, if I hydrolyze this glucose, I'm not getting any carbohydrate. Means the last single carbohydrate possible in this case scenario is glucose. Glucose cannot be further broken down into other carbohydrates. Such type of carbohydrates are called as monosaccharide. So monosaccharide is any type of carbohydrate. Yes, please go ahead and watch, because we are in a last minute for biochemistry, like FMGE. So this is going to help you a lot actually, because I am trying to cover up all the important concepts here. Okay, so it is going to be a lot of useful for you. This is a good valid question for you. Anyways, so definitely it is going to be helpful. So any sugar molecule, if you cannot break it further into any smaller carbohydrate, such type of if you cannot break into smaller version, if you cannot break into the smaller version of a carbohydrate, we'll call it as a monosaccharide. So monosaccharides are the sugars which cannot be hydrolyzed further. The classic examples include your glucose, right? You have your galactose, you have your fructose, these are the classic examples for the monosaccharides, and this is a very important point to be remembered. Why? Because they can ask you all of the following are the monosaccharides except, they can ask you. So during that time you should be able to answer monosaccharides are anything which cannot be hydrolyzed further into a smaller sugar molecules. Okay, all right, let's go to the next one, that is your disaccharide. What is that? Disaccharide. So disaccharide is a substance. If I break it, I'm going to get how many monosaccharides? Of course, two monosaccharides combined together to form a disaccharide. So basically, if at all, whenever we are seeing a scenario where what is present here, students, disaccharide. Disaccharide is such type of glucose which will give into two monosaccharides. Then you have your oligosaccharides. Which has about 2 to 10 monosaccharides, which is made up of 2 to 10 monosaccharides. So more than 10 monosaccharides, you are going to call it as a polysaccharide. Okay, so these polysaccharides can be further divided into two types. One is called as homoglycan, one more is called as heteroglycan. What do I mean by heteroglycan? What do I mean by homoglycan? Please remember one important concept here. So homoglycan is any polysaccharide, means homoglycan made out of more than 10 monosaccharide. All the monosaccharides are same. What do I mean by this? Glycogen. So if I take a glycogen is made out of glucose only. So throughout the glycogen, if you take a lot of sugar molecules are there, but all of them are made out of same type of sugar. Then we'll call it as a homosaccharide. If it is made out of same sugar. So here in this case scenario, glycogen is made out of same type of sugar. That same type of sugar is glucose. So that is why we'll call it as, what is that? Glucose, which is having a homoglycan, dear students. What is that? Homoglycan. So heteroglycans, the classic examples you have are, my dear students, glycosaminoglycans. What do we have? Glycosaminoglycan. So these glycosaminoglycans are made out of two types of sugar. What are the two types of sugar it is made out of? One is called as amino sugar. One is called as amino sugar, and one more is there acidic sugar. So is it made out of same type of sugar or different type of sugar? It's made out of different types of sugar. If it's made out of different types of sugar, then I'm going to call it as a heteroglycan. Hetero means different. So the classic examples are glycosaminoglycans, which are also called as mucopolysaccharides. What are they called as? Mucopolysaccharides. So the classic examples of glycosaminoglycans I have written here. There are some important classic examples which are there. They can ask you about their location. The location of the glycosaminoglycans are very important. Okay. So hyaluronic acid. Hyaluronic acid is a glycosaminoglycan found in synovial fluid. Found in which fluid? Synovial fluid. Synovial fluid contains which glycosaminoglycan? Hyaluronic acid. They can directly ask you, hyaluronic acid is present in which location? It is located in the, mainly in the synovial fluid. Okay. Chondroitin sulfate is present in the cartilages. Then you have your keratin. Keratin is present in the places like hair, skin, and even in nails. Okay, so what is the difference between the keratin in the nail and the keratin in the hair and skin? So keratin in the hair and skin, which is basically a type of soft keratin, we call it as soft keratin. In nail, whatever the keratin is present, a hard keratin. Okay, so sir, how keratin differs into soft versus hard? Soft keratin is something which has less number of disulfide bond. Hard keratin is anything which has more number of disulfide bonds. More number of disulfide bonds. So if more number of disulfide bonds are there, then automatically we'll call it as a hard keratin. If less number of disulfide bonds are there, then we are going to call it as a soft keratin. Then let's continue to the next one, that is heparin. Heparin is produced in the body it comes from your mast cell. Okay, so this has been a frequent question, which is a very easy question also. They'll ask you, histamine is produced from which cell? That time you should not panic. This much easy question will they ask you? Yes, it is possible to be asked about this type of easiest question also. So that time you should mark the answer as a mast cell only. Heparin is produced by which cell? Again, it is produced from the mast cell only. So there is a most important aspect, that is the next disease called as MPS. What is the name called as? MPS, called as mucopolysaccharidosis. Mucopolysaccharide. So mucopolysaccharidosis is a disease in which there is a accumulation of, in this there is what is that? Accumulation of what? Accumulation of glycosaminoglycans. So whatever the glycosaminoglycans produced in the body, they will not get digested, since due to lack of their digestion, the more glycosaminoglycan will start to accumulate in the body. Because of that, what will be there? There will be a disease called as, what is that? Mucopolysaccharide. So mucopolysaccharidosis, there are 9 types, MPS 1, 2, 3. Among which very important ones are which one? One is MPS 1, another one is MPS 2. MPS 2 is also called as Hunter syndrome, and MPS 1 is called as Hurler syndrome. Okay, MPS 1 is called as Hurler syndrome, and MPS 2 is called as Hunter syndrome. In both of them, there is a deposition of same substance, dermatan and heparin. Dermatan is present where? Dermatan sulfate is present in the skin. Okay, so dermatan and heparin sulfate will start to accumulate in these two diseases. Only difference is mode of inheritance. But rarely they had asked one very interesting question earlier, that is only MPS 9, mucopolysaccharidosis 9. So mucopolysaccharidosis type 9 occurs as a result of which disease? Occurs as a result of a syndrome called as Natovic syndrome. What is the name of the syndrome called as? Natovic syndrome. This Natovic syndrome, there is a deposition of a glycosaminoglycan, hyaluronic acid. This hyaluronic acid deposition occurs due to deficiency of what? Due to deficiency of an enzyme. So deficiency of which enzyme? Due to deficiency of a very, very important enzyme called as hyaluronase. What is that called as? Hyaluronase. Hyaluronase enzyme deficiency leads to the deposition of which one? Yes, that is due to deposition of that is only called as hyaluronic acid, which can also be named as hyaluronase, can also be called as hyaluronidase, which can be called as hyaluronase or hyaluronidase. What is the name of the enzyme? Hyaluronidase. So because of the deposition, there will be a disease that is called as Natovic syndrome. This is a previously asked question, which was a recently asked question. There is a possibility this can repeat for you in the exam. Okay, so this is a very, very important point. Let's continue to the next concept, that is dietary fibers. Dietary fibers are the complex sugars which are present in the food, but one characteristic about them is they are not digested by the body, are not absorbed by the body. Okay, so they're not absorbed, they're not being digested. So what are they helping for our body? So the main important, they reduce the risk of colon cancer, and also they increase the bulk of the stool. They increase the bulk of the stool, means it will allow easy defecation. If the food contains dietary rich fiber, that will give you relief from the constipation. So basically natural laxative, we can say. Okay, the classic examples we have are, my dear students, lignin and pectin. Lignin and pectin, these are the sugars which are complex sugars which are called as dietary fibers, not absorbed, but they are helping in only two important places which you need to remember. What are the two important places? One is in a normal individual, they increase the bulk of the stool. They increase what? Bulk of the stool. But while we are speaking in a patient who is having a high risk of colon cancer, or in a normal individual also, they're helping in reducing the risk of colon cancer. Okay, then let's go to the next one, glucose transporter. So glucose transporter are represented by a alphabets called as GLUT. GLU stands for glucose, T stands for transporter, and their location has been very important. They have asked a lot of times about the location. Every exam there is a question related to the location of the glucose transporter, so which you must remember very clearly. Let's go through that. So whenever we are speaking of a glucose transporter, a glucose transporter is a type of unidirectional transporter or bidirectional. It is a type of bidirectional. What is that? It's a type of bidirectional transporter. And the next important key point, one you must remember, so please understand, it's a type of bidirectional transporter and it works by a mechanism. The name of the mechanism is called as a ping pong mechanism. All you need to do is that remember the name of the mechanism. What is the mechanism if they ask you? It works through a ping pong mechanism. So glucose transporters, they're very important. Glucose transporter 1 is located in the red blood cell, brain, placenta. So the speciality about this glucose transporter, it does not require anybody to work. All it needs is, if glucose is present, it will work. So it is active even during hypoglycemia, and no need of insulin for its action. No need of insulin for its action. So without insulin also, glucose transporter 1 will work, and it will allow the absorption of the glucose at every scenario possible, be it hypoglycemia, be it hyperglycemia. So means very simple. Even when you are hypoglycemic, one glucose transporter is taking the glucose from the blood even at a low blood glucose level also for the survival of the body. That includes your glucose transporter 1, which is located, everybody repeat with me there only. So RBC, brain and placenta. What are the three places GLUT1 is located? RBC, brain and placenta. Okay, then let's go to the GLUT2. So if the GLUT2 is located in liver, even liver if you forget fine, but most important point you need to remember, pancreatic beta cell. Why? Because pancreatic beta cell, GLUT2 is responsible for release of insulin. Remember, release of what? Insulin. So release of insulin works with the help of which transporter, they might ask you. Release of insulin works with the, yes, of course you must remember, must remember, that is glucose transporter 2. Okay, so the most important function I want you to remember, see, there are some places where some transporter overlap in multiple organs, but this release of insulin totally depends upon the activity of one transporter in the pancreatic beta cell. That pancreatic beta cell is a glucose transporter 2. Then let's talk about the glucose transporter 3, which is located in the neurons and brain. GLUT4. GLUT4 is present in the adipose tissue, muscles, heart, and to an extent even in the liver, dear students. Even in the liver also GLUT4 is present. But remember, in all these places, GLUT4 always dependent on insulin. What is that? Dependent on insulin. Okay, so what do I mean by this? What do I mean by this is very simple. So understand, it is a bidirectional ping pong mechanism. No need of insulin is for GLUT1, does not require insulin. Insulin release depends upon GLUT2, and The function of the glucose transporter which requires insulin is always GLUT4. Means GLUT4 is nothing without insulin. GLUT4 always require GLUT4 always require insulin for its action, because insulin receptor are the one which increase the activity of glucose transporter 4. Insulin receptor, when they are activated, they increase the activity of glucose transporter 4. If insulin is not there, insulin receptors are not getting activated. Because of that, there is no activity of what? No activity of glucose transporter 4. So let's go to the next one. There is something called as a secondary active transport. There is something called as secondary active transport, that is only called as the sodium glucose transporter. Sodium glucose transporter. So basically it require two substances. One is your sodium, one is your glucose. So both of them are absorbed together or reabsorbed. If both are occurring together, then we will call it as what? We will call it as a SGLT, sodium glucose transporter. So sodium glucose transporter, dear students, it is represented by SGLT. SGLT1 is located in the intestine, especially in the small intestine. It is a typing error. It is supposed to be SGLT2 is located in the proximal convoluted tubule of the kidney. So where is SGLT2 located? Proximal convoluted tubule of the kidney. So that was just because of typing, it has gone down, that's it. But that's why I have corrected here. SGLT1 is located in the small intestine. Okay, so this is a very important concept. Let's go to the next important point to be remembered, that is your hypoxia. Whenever we are speaking of hypoxia, hypoxia can be divided into multiple types. So hypoxia means lack of oxygen supply to the tissues. Lack of oxygen supply to where? To the tissues. We call it as a hypoxia. We call it as a hypoxia. So hypoxia can be divided into types such as point number 1, hypoxic hypoxia. What is that? Hypoxic hypoxia. So hypoxic hypoxia, what happens? Okay, so please understand. Okay, so hypoxic hypoxia, there is a decrease of atmospheric oxygen, and because of that, partial pressure of the oxygen reduces. So but hemoglobin is normal, RBC is normal, red blood cells are normal, everything is normal, dear students. Simple as that, everything is normal. Only in the surrounding atmosphere there is a lack of oxygen. This occurs at a high altitude. Okay, so that time what altitude, so that time whatever you are going to develop, that will lead to hypoxic hypoxia. Anemic hypoxia is the condition where partial pressure of oxygen is normal. Okay, so there is no hemoglobin or hemoglobin is reduced. So basically, see, very simple. You have oxygen in the atmosphere that will enter into the lungs. From the lungs, that oxygen will be taken by hemoglobin. From that, hemoglobin will go to the tissues and give the supply to the tissues. So if it is giving supply to the tissues, now what you need to remember and understand here, very easy to understand. So whenever there is a lack of hemoglobin or decreased hemoglobin, means surrounding atmosphere is fine. I breathe in the oxygen, my lungs are working fine, but there is no hemoglobin to carry the oxygen from the lungs to the tissues. Because of that, my tissues are suffering hypoxia. So that time, whatever we are getting, that is only called as anemic hypoxia. The anemic hypoxia is caused due to anemia. And one more condition is carbon monoxide, because carbon monoxide will bind with the hemoglobin a lot. So carbon monoxide binds with the hemoglobin very strongly. So there is no free hemoglobin for the transport of oxygen. So that time also, similar like anemia, anemic hypoxia will develop. So carbon monoxide poisoning will lead to anemic hypoxia. The treatment is hyperbaric oxygen. So the treatment is what? Hyperbaric oxygen for the carbon monoxide poisoning. Okay, then now imagine surrounding atmosphere is fine, lungs are fine, hemoglobin is fine, but because of some problem, the blood flow is impaired. There is a problem with the blood flow. If there is a problem with the blood flow, automatically person will develop what? Stagnant or ischemic hypoxia. Basically, due to lack of blood supply, the tissues are developing a hypoxia. So everything is normal. The problem is with the blood flow. So any vascular disease, if they are causing a hypoxia, That's a classic example for what? Stagnant or a ischemic hypoxia. Okay. Then we have the next one, that is histotoxic hypoxia, which is also called as tissue hypoxia. So what is tissue hypoxia or a histotoxic hypoxia? It's very simple to understand, my dear student. Because of any problem, what has happened is very simple. There is a lack of utilization of oxygen at a tissue level. At which level? At a tissue level. Means what I'm trying to tell you is, atmosphere is normal, lungs are normal, hemoglobin is normal. The blood has gone to the from the lungs. The hemoglobin has taken the oxygen, it has gone to the tissues. Okay. Now, after going to the tissues, there is a problem with the utilization of the oxygen because of a problem with the electron transport chain. So that time what we are going to get, the hypoxia is called as histotoxic hypoxia, which occurs as a result of cyanide poisoning, even occurs as a result of carbon monoxide poisoning. Occurs as a result of carbon monoxide. So carbon monoxide causes two types of hypoxia. What are the two types of hypoxia caused by carbon monoxide? Anemic hypoxia and histotoxic hypoxia. This is a very important point to be remembered because this has been previously asked as a question. So don't make the mistake. Anemic hypoxia and histotoxic hypoxia both can be caused as a result of carbon monoxide poisoning. So since I told you about electron transport chain, so let's talk about the electron transport chain. So what is electron transport chain? So the first, let's understand the purpose of the electron transport chain, right? So the electron transport chain purpose is to produce the ATP. Purpose is to produce, what is that? ATP. So for the energy production, we have a chain called as production of ATP with the help of electron transport chain. Okay. So now when we are speaking of electron transport chain, this electron transport chain occurs in mitochondria. Occurs in where? Mitochondria. So if I take up one mitochondria, my dear students, like this, in this mitochondria, I will have two layers. I will have how many layers? Two layers. So now let's take one part of the mitochondria like this and draw it. So I will have one layer as an outer layer. One more layer will be your inner layer. So I have two layers in the mitochondrial membranes. So one is outer layer, one is inner layer. Okay. So between these two layers, there is a space called as the intermembrane space. There is a space called as intermembrane space. Okay. So this intermembrane space and these ones, let's understand very simply. So on the inner membrane, on which membrane? On the inner membrane, we have something called as complexes. What are they called as? Complexes. Okay. So complexes are what? Basically, they are the enzymes. What are they? They are the enzymes. So on the inner mitochondrial membrane, what we have is enzymes. Okay. And along with that, there is also something called as transporters. So there are two things which you need to remember: transporters, and also we have complexes called as, basically they are the enzymes. So whatever the glycolysis, whatever the Krebs cycle, whatever the oxidation of the fat oxidation or glycolysis or Krebs cycle, during all these cycles, we are getting energy in two forms. One form is called as ATP. फिर से बताता हूँ, ध्यान से सुनो. During all the oxidative processes such as glycolysis, such as oxidation of the fats, such as Krebs cycle, during this time we are getting energy. How we are getting energy? In two forms. One is in the form of hydrogen. One more is in the form of ATP. ATP can be directly used as energy. Now, whatever the hydrogen, whatever is there, which is coming from the glycolysis, let's imagine glycolysis. During the glycolysis, there was some hydrogen which was released. So this hydrogen will be taken by somebody that is called as the hydrogen acceptors. What are they? Hydrogen. These hydrogen acceptors are your NAD and FAD. Okay. After taking the hydrogen, NAD will become into NADH. After taking the hydrogen, FAD will be coming to FADH. So all this hydrogen acceptor taken the hydrogen. After taking the hydrogen, what will happen? This will go and enter into electron transport chain. So when this NAD comes here, so from the NADs, the complex number 1, the name of the complex is called as the NADH dehydrogenase. NADH dehydrogenase. So basically all the complexes are what? Enzymes. All the complexes are enzymes. So NADH dehydrogenase will extract the hydrogen and also extract the electron. Basically there is an enzyme that is called as NADH dehydrogenase. So that will break the NADH. When it breaks the NADH, we are getting hydrogen and also we are getting few electrons. All these electrons obtained from where? From the complex number 1. Point number 1 is from the complex 1 you have electrons and also hydrogen was produced. Whatever the hydrogen is produced with the help of the same complex number 1, all the hydrogen will come into the intermembrane space. So basically in the electron transport chain, so far what has happened? NADH dehydrogenase came, okay, which separated the hydrogen and electrons from NADH. All the hydrogen was sent into the intermembrane space and all the electrons were given to somebody called as coenzyme Q. What is the name of the enzyme? Coenzyme Q. Okay, so which is also called as ubiquinone. What is that called as? Ubiquinone. Ubiquinone is also called as coenzyme Q. So this ubiquinone is a type of transporter. What it is transporting? It is taking the electrons. Okay, so far from the NADH, hydrogen and electrons are separated. Hydrogen is sent to the membrane space, electrons are given to the coenzyme Q, and the story over with the NADH. Now let's go to the next one, complex number 2, which is called as the FADH dehydrogenase. It will also break the FADH into hydrogen and electrons. So all the hydrogen either with the help of complex 3 or with the help of complex 1, because look here, complex 2 is not interacting with the intermembrane space. The answer is no. The answer no. It is not interacting with the intermembrane space. So if it is not interacting with the intermembrane space, so automatically other complexes will then send the hydrogen into where? Into the intermembrane space. So all the hydrogen was sent into the intermembrane space and all the electrons from the complex 1 and from the complex 2, the electrons are given to the coenzyme Q. This coenzyme Q will give the electrons to the complex number 3. From there, all the electrons will go to the next transporter called as the cytochrome. Cytochrome C. Cytochrome C is an enzyme or a type of transporter. Where it is taking the electrons from where to where? From the complex 3, it is giving to the complex 4. So now the name of the complex 3 is only called as cytochrome reductase. The name of the complex 3 is called as cytochrome reductase. The next complex is called as cytochrome oxidase. Cytochrome oxidase is an enzyme which will take all the electrons, and does the reaction oxidation. Which reaction? Oxidation. And while undergoing the oxidation, whatever the oxygen which came from the breathing, all the oxygen will be utilized. So cytochrome oxidase utilize the oxygen. So basically, all the oxygen molecular level oxygen utilization is done by the enzyme cytochrome oxidase, which is also called as complex 4. So during this oxidation, it will require the electron. That electrons are coming from the complex 1, 2, from there to coenzyme Q, then to the complex 3, from there to the cytochrome C, from there it has gone to the complex 4. So basically, all the electrons are transported towards the complex 4. So all the hydrogen was coming where? All the hydrogen from all the complexes will come into the intermembrane space. Now all this hydrogen will go towards the complex number 5. The name of the complex is called as ATP synthase. The name of the enzyme is called as ATP synthase. So basically this ATP synthase, my dear students, it does one reaction called as the phosphorylation. Which reaction? Phosphorylation. So there are two reactions which are simultaneously occurring in the electron transport chain. One is your oxidation, one more is your phosphorylation. So always remember, oxidation and phosphorylation are a coupled reaction. Oxidation and phosphorylation are your type of coupled reaction, or your which type of reaction? Coupled reaction, or your type of which reaction? Coupled reaction. So if this coupled reaction occurs, only then we are getting a ATP. For the synthesis of ATP, what is required? Both the processes to occur. Which are those? That includes your oxidation and phosphorylation both together. We call it as a oxidative phosphorylation. We call it as what? Oxidative phosphorylation. So if there is a oxidative phosphorylation, so oxidative phosphorylation can be uncoupled due to some poisons. So uncouplers and complex inhibitor might make a confusion to you people. So I want you to clearly listen to me very clearly. Uncoupler separate these two reactions. Uncouple the oxidative phosphorylation. If there is an uncoupling of the oxidative phosphorylation, it will generate only heat, but there is no energy. There is no energy. Remember, it's simple as that. Like, you might have seen these type of scenarios maybe. Okay. So whenever two people are together happily, ठीक है, वहां पे कोई ना कोई होता है छेड़छाड़ करने के लिए. There will be someone to separate these two people. Okay, that is only in the name of bestie, right? Like that only these uncouplers happily it was occurring as a coupled process. These uncouplers will come and separate them both. Once they separate them both in real life, once two people are separated due to third person, that whole situation will become very hot, very complicated. Like that only, when this uncoupled body will produce only heat, there is no generation of ATP. So uncouplers increase the heat production rather than producing the ATP. Okay, so that uncouplers, the classic examples we have are 2,4 dinitrophenol. 2,4 dinitrophenol. Drugs such as aspirin, hormones such as thyroid hormones, and there is one more chemical called as thermogenin. The chemical that is thermogenin, it is found in the brown fat in the infants. It is found in the brown fat in the infant. Actually, it is helpful in infants. Why? Because infants are more prone to hypothermia. Infants are very prone to hypothermia. So to protect them from the hypothermia, the body itself has a chemical. The name of the chemical is called as the thermogenin, which is found in the brown fat. Right. So this is a story of your, what is that? A story of your, yes, story of your, very importantly, uncouplers. Very importantly, uncouplers. So once you understood about the uncouplers, let's go to the next concept, that is only inhibitors of electron transport chain. So basically they inhibit the enzymes of the electron transport chain, and this also has become a very commonly asked question. So remember, so there are some chemicals such as barbiturates and rotenone. Barbiturates and rotenone. So barbiturates and rotenone will inhibit complex number 1. Malonate will inhibit complex number 2. Dimercaprol and antimycin will inhibit complex number 3. Carbon monoxide and cyanide poisoning will block the complex 4, among which this is a complex 4 inhibitors have been a more important than other ones. But remember this, once if you go through, you can remember this very easily. Complex 5 inhibitor is oligomycin. Oligomycin. Okay. Hope everybody able to understand. Hope everybody able to understand. Okay. So let's continue. The next one is glycolysis. Next pathway is which one? Glycolysis pathway. Which pathway? Glycolysis pathway. So when we are speaking of glycolysis pathway, so please listen to me. Glycolysis are also called as an Embeden-Meyerhof pathway. So this is a pathway in which breakdown of glucose to pyruvate. Glucose to what? Pyruvate. Glucose to what? Pyruvate. So the conversion of glucose to pyruvate, we call it as, that is only glycolysis. So when we are speaking of glycolysis, so initially whatever the food you ate, from the food glucose was extracted. All the glucose went into where? All the glucose went into the cells. In the cell, glucose is a 6 carbon containing substance which will undergo the influence of an enzyme called as the hexokinase. But in the liver, liver, we have the same isoenzyme, means which does the same activity. The name of the enzyme is called as glucokinase. As a result of glucokinase, the glucose will get converted into glucose 6 phosphate. So we are using one ATP. We are using one ATP and adding a phosphate group into the glucose at a 6th carbon position. At which position? 6th carbon position. So that is why we call it as a glucose 6 phosphate. Now this glucose 6 phosphate, whatever is there, under the influence of a isomerase, what is the name of the enzyme? Isomerase, it will give rise to fructose 6 phosphate. Now fructose is all isomer of glucose. So we are getting, instead of glucose 6 phosphate, under the influence of isomerase, we are getting fructose 6 phosphate. This fructose 6 phosphate again under the influence of kinase. Kinase, wherever the enzyme name comes, we are transferring a phosphate group from one molecule to the another molecule. Means here we are transferring the phosphate from the ATP into the fructose. So now it will become into fructose 1 comma bisphosphate. Fructose 1 comma 6 bisphosphate, fructose 6 phosphate, everywhere it is 6 carbon only. Okay, now this fructose 6 phosphate will break itself into two substances. One is called as dihydroxyacetone phosphate. Dihydroxyacetone phosphate. And one more substance is called as glyceraldehyde phosphate. Glyceraldehyde. Now, one molecule of dihydroxyacetone phosphate, one molecule of GLA. Now this one molecule dihydroxyacetone phosphate is there, right? This will also get converted into GLAP. So totally I will have how many GLAP? 2 GLAP. Why 2 GLAP? One directly comes from the fructose 1 comma 6 bisphosphate and one more is coming from the DHAP, because DHAP will further get converted into GLAP only. This PDF will be uploaded in the description section. Tonight itself PDF will be sent. Our team will upload it. Okay. So two molecules of GLAP will get converted into 1,3 bisphosphoglycerate. So during that time we are getting, so two molecules is breaking down into two molecules of 1,3 bisphosphoglycerate. So we are getting what is two molecules of 1,3 bisphosphoglycerate. So it will generate two molecules of NADH. How many molecules of NADH we are getting? Two molecules. Okay. Then this 1,3 bisphosphate will get converted into 3 phosphoglycerate. So during that time, it is eliminating 2 phosphate groups. So we are getting 2 ATPs here. So this 3 phosphoglycerate to 2 phosphoglycerate, again 2 times it will reaction occur. Why 2 times reaction occur? Because from here, from GLAP, everything is occurring 2 times. That's it. That's why here we have 2 NADH, 2 1,3 bisphosphoglycerate, 2 molecules of ATP, 2 molecules of 3 phosphoglycerate. This will get converted into 2 phosphoglycerate. Okay, this will get converted into phosphoenol pyruvate. This phosphoenol pyruvate, under the influence of pyruvate kinase, it will give rise to 2 ATP and we are getting 2 pyruvate. So 1 glucose is equal to 2 pyruvate. 1 glucose is equal to 2 pyruvate. Okay, I hope this is clear. 1 glucose is equal to 2 pyruvate. So this glucose to this conversion will lead to what is that production of your, yes, that is pyruvate. So there are some important points to be remembered. So energy, 1 glucose to pyruvate, what happened? Let's see. NADH, how many we got? Everybody tell me, NADH, how many we got? 2 NADH. 2 NADH is equal to 5 ATP. Right? 2 NADH is equal to 5 ATP. So here we got 2 ATP, here we got 2 ATP. As the ATP itself, we generated 4 ATP. So total number of ATPs we are getting from glucose to pyruvate is 9 ATP, but 9 ATP is the gain. Okay, but how many is the loss? So let's look into the cycle. First, here we used 1 ATP, here we used 1 ATP. So total loss ATP is 2 ATP. So this 2 ATP minus if we do, so automatically we are getting 7 ATP, 1 glucose to net gain of ATP from 1 glucose to pyruvate conversion. So during that time, we are getting how many ATP? 7 ATP in a aerobic glycolysis. The same thing if it is an anaerobic glycolysis. So in the anaerobic glycolysis, this NADP Two NADH is there. Na? So in the anaerobic glycolysis, NADH is utilized. NADH is utilized. So how many NADH? 2 NADH is utilized. So basically we are not getting this 5 ATP. Only we are total getting 4. 4 minus 2 will become 2 only. So what I am trying to tell you is very simple. So in a aerobic glycolysis, net gain will be 7. Okay. But in anaerobic glycolysis, whatever NADH produced in the cycle, that NADH will also be utilized also. NADH will also be utilized. So if NADH is utilized, so the net gain ATP in the anaerobic glycolysis is 2 ATP only. That is why aerobic glycolysis is very important, because it gives you more amount of ATP. That is, aerobic glycolysis give you 7 ATP. Anaerobic glycolysis will give you a very small amount. How much? 2 ATP only. So let's go to the next important rate limiting enzyme. The rate limiting enzyme is phosphofructokinase. The rate limiting, the concentration of which enzyme controls the complete activity of glycolysis. That glycolysis is controlled by, of course, that is by your phosphofructokinase. This phosphofructokinase also have isoenzyme alpha and beta, which can also be called as the PFK1 and PFK2. It's the same thing, but you guys have to remember what is the rate limiting enzyme. You don't have to remember alpha beta. At least you got to remember what is that. Phosphofructokinase is the rate limiting enzyme of glycolysis. Okay, so glycolysis occurs in almost all the tissues. In which part of the tissue? This is going to be a very important point. In the cytoplasm. In the cytoplasm. So cytoplasm is the place where there is a glycolysis which is occurring in almost all the tissues. So now please remember hexokinase. In the starting itself, I told you about one isoenzyme. Remember, that is in the liver hexokinase. Okay, sorry, in other tissues, the exokinase. In the liver, the same action is done by glucokinase, right? So in the liver, in the liver, the enzyme which is present is glucokinase. Insulin will induce this enzyme. Means if at all a person has more amount of insulin released, or for example, whenever insulin is released in the body, that time what happens to the glycolysis in the liver? It will increase, because hexokinase activity will be more. Glucokinase. In the liver, we have isoenzyme glucokinase that will be increased. The activity will be increased as a result of insulin. So if it is induced, automatically there is an increase of glycolysis in the liver. Increase of glycolysis in the liver. So increase of glycolysis in the liver is done by glucokinase, while as hexokinase has it present in all the tissues, but insulin has no action. Insulin has no action. Insulin has no action on what? On the hexokinase. Okay. So let's go to the next one. Pyruvate kinase deficiency. So remember, in the red blood cell, pyruvate kinase is an enzyme which is a last step in the aerobic glycolysis, right? So if this enzyme is not there, automatically, look here, there are 2 ATPs which is coming from here. So please understand, pyruvate kinase deficiency in the red blood cell, if that is lack, automatically there is a decrease of ATP. So why there is a decrease of ATP? Remember, this 2 ATP which is being generated, that will not be generated. Point number 1. Point number 2, you might be thinking, sir, still we have NADH, right? That NADH may be used for the energy production. No, we cannot use NADH, because in the red blood cell there is no mitochondria. NADH utilization requires electron transport chain. Electron transport chain occurs in the mitochondria. RBC has no mitochondria. NADH is of no use in red blood cell. And already here we are losing 2 ATP because of lack of this enzyme pyruvate. So totally we will get only 2 ATP from here, and 2 ATP already it will be utilized. One is here, one is here. So in the RBC, in the red blood cell, normally anaerobic glycolysis will occur. But if pyruvate kinase is not there, that anaerobic glycolysis is also impaired. If anaerobic glycolysis also impaired, automatically pyruvate kinase deficiency leads to an anemia called as the hemolytic anemia. Okay, so this is having a autosomal recessive inheritance. So that is the story of your pyruvate kinase deficiency. Then In the Krebs cycle, the most important point to be remembered is so rate limiting enzyme for TCA cycle is isocitrate dehydrogenase, and TCA cycle occurs in cytoplasm or mitochondria. It occurs in the mitochondria. It occurs in the mitochondria. Okay, let's go to the next disease. It is a disease associated with impaired metabolism of fructose, that is HFI. What is this? HFI, hereditary fructose intolerance. So very simple to understand hereditary fructose intolerance. So because since it is a very quick rapid revision, I'm just going through the important clinical aspects here. Okay, but just give you an idea, just let me give you a simplified version of fructose metabolism. So you have fructose from the food. Okay, that fructose undergoes the influence of an enzyme called as fructokinase. What is the name of the enzyme? Fructokinase. This fructokinase convert the fructose into fructose 6 phosphate. This fructose 6 phosphate will get further converted into fructose 1,6 phosphate. This fructose 1,6 phosphate like glycolysis only, but one small difference is one enzyme is there, aldolase B. The name of the enzyme is aldolase B. Okay, so because of the aldolase B, it will break itself into GLAP and DHAP, so which is basically similar to that of glucose metabolism, So if there is a lack of one enzyme called as aldolase B, which enzyme is deficient? Aldolase B. Because of which gene mutation? Gene mutation is ALDO-B gene mutation, which is an autosomal recessive disorder. It is caused by the ALDO gene B mutation, which will lead to aldolase B deficiency, that will lead to a disease called as HFI, which is called as hereditary fructose intolerance. These people will develop hypoglycemia after consumption of fruit juice. After consumption of fruit juice, there will be severe vomiting. Okay, and also recurrent episodes of hypoglycemia are possible. All right. Then the next one, if there is a deficiency of fructokinase, so fructose will come into the body, but fructose will not get converted into fructose 6 phosphate. So all the fructose which is absorbed in the blood will start to enter inside the urine, because you ate the fructose got absorbed from the GIT. Now it has gone into the blood. In the blood, fructose level is elevated, but fructose is not getting metabolized due to lack of fructokinase. So this remaining fructose will enter into the urine, leading to essential fructoseuria. This is a unmetabolized fructose in the urine. It causes the elimination of a unmetabolized fructose in the urine. This type of condition, we call it as what? We call it as essential fructoseuria. Then let's go to the next one, galactose metabolism. Okay, what is the metabolism? Galactose metabolism. It is due to deficiency of one enzyme called as galactose 1 phosphate uridyl transferase, which is also called as GALT. Galactose 1 phosphate, okay, uridyl transferase deficiency will lead to a condition called as galactosemia, which is an autosomal recessive disorder, which is typically characterized by oil drop cataract. Okay, so the main clinical feature is oil drop cataract, maybe developmental delays and intellectual disabilities, which can be overlapping with the other clinical feature, but oil drop cataract is a very, very important associated feature of your, what is that? Of your galactosemia. Okay, then let's go to the next condition. What is that? That is your glycogen storage diseases. So glycogen storage diseases, the very, very important, highly important topics, my dear student. Glycogen storage disorder is a highly important topic. So let me quickly take an image for the explaining the glycogen storage diseases, because this part is going to be like right now, you, even if you can't remember the whole metabolism of glycogen, that is not going to affect you, but the problem is just you need to have an idea what is happening with the glycogen. Once if you have an idea what is happening with the glycogen, it is going to be easy for you to understand and remember the disease names. So for that purpose here, I have one image. Let me quickly explain about the glycogen storage so that we can understand the diseases. Okay. So we have a glycogen, how glycogen synthesis occurs. Let's understand. See, first of all, let me explain the glycogen. Glycogen is a polymer of glucose. Glycogen is a polymer of glucose. Polymer of glucose. So by glycogen is a polymer of glucose means glycogen is made out of glucose. It is a type of homoglycan. It's a type of homoglycan. It's a type of homoglycan. It is made out of only one type of sugar. What is that one type of sugar? That one type of sugar is glucose. So when you take a structure of the glycogen, so in the center we'll have one protein called as glycogenin. With this glycogenin, so all these are lots and lots of glucose will be accumulated. Lots and lots of glucose attached to each other. Lots and lots of glucose is attached to each other. So if you can notice here like this, okay. So if I take out, so lot of what is there? Glucose. So all these small dots which I'm representing with the blue, this is basically I'm talking about a glucose. So many glucose attach with each other. So this attachment here we are having which type of one? It is a straight chain. It's a linear chain. So linear chain wherever it is there, that will create a 1,4 bond. Means one glucose binds with another glucose by a bond called as glycosidic bond. Which glycosidic bond? Alpha glycosidic bond. Okay. So if it is in a linear chain, we call it as 1,4 bond. Basically, for first carbon of this glucose and fourth carbon of this. But in some places, if you notice here, if you notice, there is branching which is occurring. Yes. So at the branching level, we have 1,6 bond. 1,6 bond. So we have a 1,4 bond and we have a 1,6 bond. So 1,4 is a type of bond. What is this 1,4 bond? It is present in a linear chain. So 1,6 bond is there. That 1,6 bond is in a linear chain. So whatever the excess glucose is there, that excess glucose will get converted into glucose 6-phosphate. This glucose 6-phosphate will get converted into glucose 1-phosphate. This glucose 1-phosphate will get converted into UDP glucose. So this UDP glucose, lots and hundreds of UDP glucose undergoes the effect of one enzyme called glycogen synthase. What is the name of the enzyme? Glycogen synthase. Under the influence of glycogen synthase, the linear chain, the straight chain is formed. This 1,4 bond chain is formed as a result of what? Yes, as a result of glycogen synthase. But this long chain cannot be made because it will become very lengthy. So now what we should do? Our body has to make the branches. The branching is done by your branching enzyme which forms 1,6 bonds. Which forms 1,6 bond. So after branching and the straight chain, what we are getting is glycogen. But whenever from the glycogen our body wants glucose, glycogen will break into glucose 1-phosphate and a limit dextrin. So the straight chain, the straight bond is broken by glycogen phosphorylase, but the branching areas are broken by branching enzymes. Sorry, debranching enzymes. So if you want to make a branches, we require branching enzyme. If you want to break a branch, we require debranching enzyme. Okay. So this glucose 1-phosphate will get converted into glucose 6-phosphate. This glucose 6-phosphate under the influence of glucose 6-phosphatase, it will get converted into glucose. So we started from the glucose, we got the glucose back. That's what is occurring. Okay, so let's continue. Because of any of the defect in any of these enzyme, person can develop a disease called as glycogen storage disease. Okay, so the type 1 glycogen storage disease, it is only called as Van Giereg's disease. What's the name of the disease? Van Giereg's disease. Van Giereg's disease occurs as a result of glucose 6-phosphatase deficiency. Okay, the common clinical feature include hepatomegaly and hypoglycemia. But these hepatomegaly, I'm telling again and again, many students get confused. Hepatomegaly, hypoglycemia might be associated with other conditions also. It is possible to have, but hepatomegaly and hypoglycemia more commonly associated with Van Giereg's disease, but it can be associated with other conditions also possible. Now, Pompey's disease type 2. Pompey's disease occurs as a result of acid maltase deficiency. Acid maltase is present in the lysosomes. Okay, so excessive glycogen will enter the lysosome. In the lysosome, this glycogen has to be broken by acid. But if that if acid maltase is not there, now what will happen? More glycogen will enter into the lysosome, but in the lysosome there is nobody is there to digest. Who was supposed to be present? Acid maltase. If acid maltase is not there, automatically in the glycogen, these glycogen will get stored in the lysosomes. Okay, so it is a type of lysosomal glycogen storage disease which predominantly affects your muscles and heart. Predominantly affects what? Muscles and the heart are predominantly affected. Okay, so that is your cardiomyopathy, hypotonia are commonly seen with Pompe's disease. Then we have Cory's disease. Cory's disease is due to deep branching enzyme deficiency. Then we have Anderson disease. Anderson is due to branching enzyme deficiency. Then we have McArdle's and Hirsch disease. Both of them occur as a result of glycogen phosphorylase only. Okay, but the difference is very simple. McArdle's occurs in the muscles. In the muscle, if glycogen phosphorylase is defective, we are getting McArdle's. But if it is same thing which is defect in the if it is defective in the where, if it is defective in your, yes, very importantly, if it is defective in the liver, that is hepatic glycogen phosphorylase deficiency leads to Hirsch disease. Okay, so then there is one more disease, that is type 7 is there. Type 7 is called as Tarus disease. Tarus disease. So Tarus disease occurs as a result of what? Occurs as a result of deficiency of phosphofructokinase. Phosphofructokinase. Okay. So this is the story of your, what is it? Story of your all the carbohydrate related points and glycogen storage disorders. So there is a high likely they can ask you one important question possible from the glycogen GST, glycogen storage diseases. Okay. So now take a 10 minutes break and we'll continue. 10 minutes. All right, everyone, come back. Let's start. So just let me open the chat box. So far what we discussed was mainly about the carbohydrates. Now let's continue into the other important aspects of the discussion. Okay. Perfect. So what we have here so far was carbohydrate. What's a very important topic? As I told you, all the important previous year topics also have been covered, and also I have told you what are the possible ways of questions which can be asked. So in this mainly what can be asked, I'll tell you very importantly, dear students, that is you need to remember one thing. What is that? That is these enzyme deficiencies which will lead to the diseases are very important. Okay, so once we understood about that, especially this glycogen storage disorder, every year there is a possibly they have asked many times they have asked these questions. Okay, so you might be thinking, sir, these are rare diseases. Though they're rare diseases, very confusing diseases, so that makes a very good point for asking you the MCQ based questions. Okay, so once we understood about that, let's continue to the next condition, that is G6PD deficiency. So G6PD, what is G6PD? That is only called as an enzyme called as a glucose 6 phosphate dehydrogenase. What is the name of the enzyme? Glucose 6 phosphate glucose 6 phosphate dehydrogenase. So when we are speaking of glucose 6 phosphate dehydrogenase, where is this enzyme importantly located, in which pathway? It is a rate limiting enzyme. It's a rate limiting enzyme of HMP pathway. It's a rate limiting enzyme for HMP pathway. Okay, so G6PD deficiency, it's a very interesting point to be remembered. Why? Because understand, so this G6PD, whatever is there, this G6PD will produce one substance called as NADPH, not NAD, NADPH, my dear students. What is that? NADPH. Now this NADPH is a very important substance for the survival of RBC. How is it important for the survival of RBC? So remember one important key point, that's enough. It is responsible for the removal of H2O2. What is it? H2O2 from the RBC. It is responsible for what? Removal of H2O2 from the RBC. What is H2O2? H2O2 is called as ROS, that is reactive oxygen species, which will produce the free radical. Reactive oxygen species which produce the free radical. Okay, so every time remember, free radicals are one of the source for the free radical is H2O2. That H2O2 is a reactive oxygen species. Okay, so this is a type of X-linked recessive disorder in which what happens is that there is a decrease of NADPH. So if there is a decrease of NADPH, automatically there is an increase of H2O2 in the red blood cell. So if there is an increase of H2O2, that is hydrogen peroxide, that will produce the free radical. That will produce which radicals? Free radicals. As it produces the free radical, this free radical destroy the red blood cell, causing a hemolysis, and also it creates one of the most important bodies called as Haynes bodies. Which bodies? Haynes bodies. Haynes bodies are produced from the RBC cell membrane as a result of a reaction with the hydrogen peroxide. So this is only called as Haynes body, is a lipid peroxide body. It's called as lipid peroxide body. So once this lipid peroxide body is formed, that lipid peroxide bodies are removed by the spleen, so creating a bite cell. So bite cells are produced as a result of removal of the Haynes body by your splenic macrophage. So the patient will have a features of hemolytic anemia. And remember the very important, normally G6PD deficiency children throughout their life, they can remain healthy. But only during the time of excessive free radicals in the body. So excessive free radicals in the body, any condition which increase the free radicals will precipitate the disease. Means the child will remain healthy. When they are exposed to this precipitating, means triggering factors, that time they will develop a hemolytic anemia. Which anemia? hemolytic anemia. So precipitating factors are your, what is that? Precipitating factors are your older anti-malarial drugs such as hydroxychloroquine, mefloquine. These and all your older anti-malarial drugs. Okay, then if the child has the inflammation, maybe respiratory tract infection, urinary tract infection, maybe there is a fever, during that time also the free radical production will be increased, because in pathology you would have read inflammation is one of the sources for the free radical, and sulfat drugs will also trigger the G6PD deficiency, and after the consumption of fava beans also. So during, see, for example, the question will come like this. A child develops a hemolytic anemia like clinical feature after getting treatment for a malaria. So after getting treatment for a malaria, he developed a hemolysis like clinical feature. It is a classical indicator of G6PD deficiency. So G6PD deficiency, if it is there, the child will develop hemolytic anemia. So these precipitating factors are your, what you answer? Old anti-malarials, inflammation, and sulfur drugs. Okay. So let's go to the next important. The gold standard investigation is G6PD assay, but in the peripheral smear you are going to find the white cells, and even you can find the Heinz bodies. Heinz bodies are lipid peroxide bodies. These lipid peroxide bodies will be removed by the splenic macrophages, creating the white cells. Let's go to the next important chapter. What is that next important chapter, my dear students? That is only your very important one. That is your, yes, that is your amino acid. Amino acid. So there is a simple classification and important key points to be remembered. Amino acid. Amino acid should contain two important what is that? Two important functional groups. One is ammonia group. One is your COOH group. COOH group and ammonia group together, they are going to form your, what is that? Amino acid. Means in a amino acid, two functional groups are there. These two functional groups are NH3 and COOH. Okay, so amino acid without any chiral carbon is glycine. What is a chiral carbon? Any carbon atom which is attached to the four different groups, because carbon valence is 4. Imagine it is attached to four different substances. Okay. So one carbon is there, it is attached to four different substances. If it is attached to the four different substances, we'll call it as a chiral carbon. So chiral carbon, so which amino acid does not have a chiral carbon? That is your glycine. Glycine is the simplest amino acid. Okay. Amino acid. So very simple. So I told you people, let's imagine the structure of an amino acid. This is the R group, C carbon is there, COOH and ammonia. So this is a type of, what is that? NH3. It's a type of course, it's a type of your amino acid. If this amino group is freely not there, means it is attached to somebody. Let's imagine it is attached to a ring. Basically, if amino group is attached with somebody, then we call such type of amino acid as a amino acid. Means amino group is not free. The classic example is proline. So they'll ask you which of the following is the example for the amino acid. The example for the amino acid is your which one? That is your proline. So one amino acid bind with each other to form, what is that? With another amino acid. Like that, many amino acid combine together to form a protein. Okay. So amino acid attach with each other with the help of peptide bond. That is why proteins are called as polypeptides. Why do we call them as polypeptide? The polypeptides are called because of the peptide bond. Okay. So the next important concept, amino group of one amino acid form a peptide bond with carboxyl group of another amino acid. I told you I have one amino acid here, another amino acid here. Both are attached with each other. If both are attached with each other, one amino acid binding with the another amino acid. So where they are binding, one amino acid's amino group binds with the COOH group of the another amino acid. Okay, so that is the location of your peptide bond. So classification of amino acid, simplest amino acid are glycine and alanine. Branched chain amino acids are three of them are there: leucine, isoleucine, and valine. It can be remembered with a mnemonic live. It can be remembered with a mnemonic live. Okay. Sulfur containing amino acids we have methionine and cysteine. Hydroxyl containing amino acids are serine and threonine. Serine and threonine are a hydroxyl containing, means they contain OH group. Okay, so then hydroxyproline in collagen and desmos in elastin are derived amino acids. What is a derived amino acid? Normally, all the amino acids are coded by codons. For example, methionine is coded by a codon called as AUG. Coded by a codon called as AUG. So, like that, every amino acid should be coded by some codon. AUG, ACG, AAA, something like that, right? So if amino acid is not coded by any codon, that type of amino acid, we'll call it as a derived amino acid. The classic example is not proline. Proline is coded by the codon, but hydroxyproline is not coded by any amino acid. We call this type of amino acids as a derived amino acid. And we have 21st amino acid, that is your selenocysteine, and 22nd amino acid, that's your pyrrolysine. Okay, so here the next cycle we are going to discuss is urea cycle. So just let me take up the image. There is small typing mistakes in that image. Let me clarify that one. Let me get the different image. Okay. So when we are speaking of urea cycle, my dear students, occurs where? Urea cycle occurs in both cytoplasm and mitochondria. So which part of the body? It will occur in the liver. In the liver, in the mitochondria, all the waste product ammonia comes to the liver. All the ammonia combines with carbon dioxide in the mitochondria, creates substance called as carbamyl phosphate with the help of an enzyme called as the CPS1 enzyme. With the help of an enzyme called as CPS1, carbamyl phosphate synthase 1. What is that? Carbamyl phosphate synthase 1. This is the rate limiting enzyme of your, what is that? Urea cycle. So this carbamyl phosphate combines with ornithine, which creates something called as citrulline. With the help of an enzyme called as the ornithine transcarbamylase. So this ornithine transcarbamylase will give rise to citrulline. Citrulline will further give rise to arginosuccinate, will give rise to arginine. From the arginine, you are getting urea and ornithine. So basically, the amino acid which breaks itself into give rise to urea is your arginine. The location of urea cycle is in liver, and most importantly, the enzyme responsible for is arginosuccinase and ornithine transcarbamylase, that is OTC, and CPS1 are the initial enzymes. In this, CPS1 is the most important enzyme, that is a rate limiting enzyme. Okay, so they will ask you one most important this thing. Okay, what is that? That is rate limiting enzyme. They'll ask you which amino acid responsible for the urea production. That's your arginine. Okay, so disorders of urea cycle will lead to a disease called as a hyperammonemia. So if there is a lack of this enzyme, that is only carbamyl phosphate synthase, automatically ammonia will not converted into urea, leading to excessive ammonia accumulation. We call it as a hyperammonemia. Hyperammonemia occurs as a result of deficiency of one enzyme. The name of the enzyme is called as CPS1. In CPS1, there is an increased level of ammonia. Okay, and OTC, ornithine transcarbamylase deficiency, leads to type 2 hyperammonemia, in which there is an increase of ammonia and also there is an increase of uric acid. Because look here, if there is a lack of this enzyme, that is ornithine transcarbamylase, so automatically carbamyl phosphate level will increase. This carbamyl phosphate, if it is increased, it enters into a alternate pathway. Leading to what is that? Leading to elevated level of uric acid. Okay, so the next amino acid what we have, my dear student, that amino acid is only your, yes, that is